Fluorescent probes now let researchers map water-transporting xylem in living plants with greater speed and specificity than older dyes. This is Planetterrian Daily, episode 139. In the past week, we saw nanoplastics reaching neonatal mouse brains with distinct spatial patterns, a spatiotemporal map tracing how tumors spread to distant sites, and a new genetic tool that maps how cells choose which of their 20,000 genes to activate at any moment. Threads that continue to highlight how environmental exposures, disease progression, and gene regulation intersect. A new atlas of lysosomal aging maps metabolite changes that overlap with those seen in lysosomal storage disorders. The work draws on Science journal findings to link age-related shifts in lysosomal function with disease-like metabolic patterns. Researchers compiled data across cell types and ages to reveal consistent signatures. This approach highlights how lysosomal decline contributes to broader cellular aging processes. Follow-up work will test whether the signature can serve as a biomarker in human cohorts. A few episodes back, it was cellular aging mechanisms. Today's atlas adds the metabolite signature linking lysosomal decline to storage disorder patterns. Now, shifting to efforts that aim to build replacement tissues across species. Institute of Science Tokyo researchers identified xenophagocytosis by embryonic macrophages as a barrier to donor cell survival in rat to mouse chimeras. Blocking this response increased donor cell persistence and improved generation of rat pancreas tissue. The strategy targets a natural immune clearance process rather than broad immunosuppression. Results bring functional organ generation across species closer to practical use. Next experiments will test additional donor-recipient combinations and refine the blockade method. The targeted intervention raised survival of rat donor cells inside mouse embryos and produced more complete rat pancreas tissue. On a different note, evolution reuses existing neural pathways for new behaviors. In clonal raider ants, two chemical signals toggle between foraging and larval care, with the balance shifting as workers age. The same signals operate in mammalian caregiving systems. The study suggests evolution reused ancestral hunger control pathways for new social functions. Findings were obtained through targeted manipulation of the signals in ant colonies. Future work will examine whether similar repurposing occurred in other social insects. Most people assume parenting behaviors evolved as entirely new brain programs built from scratch. In reality, the same chemical signals that once drove foraging and hunger responses now also control whether an ant cares for larvae or leaves the nest. Right now, as clonal raider ants age, the balance between those two signals shifts, flipping the animal from one task to the other without requiring a separate neural architecture. The study shows the signals are chemically related to pathways that also operate in mammalian caregiving, indicating deep conservation across distant species. One memorable detail is that a single pair of signals can produce two opposing behaviors simply by changing their relative strength over time. The practical takeaway is that interventions aimed at social or caregiving deficits in humans may be able to target existing circuits rather than needing to create new ones. Watch for future work that maps whether similar repurposing explains other complex social behaviors across vertebrates, from neural repurposing in insects to new tools for visualizing fluid transport in plants. Researchers at the Indian Institute of Technology, Gandhinagar, and the Regional Center for Biotechnology developed a new class of fluorescent probes that stain xylem tissue faster and with higher specificity than conventional dyes. The method improves visualization of water and mineral transport structures in intact plant samples. Published in Plant and Cell Physiology, the work targets vascular biology questions relevant to crop resilience. The probes reduce background staining and increase sensitivity, allowing clearer observation of developmental changes in xylem. Next steps include testing the dyes across additional plant species to support broader studies of vascular function. From plant vascular imaging to broader microbiome shifts that accompany aging, a study reports a direct association between shifts in gut microbial communities and markers of aging. The work examined microbial composition alongside physiological parameters across age groups. Specific taxa showed consistent correlations with age-related decline. The findings add to evidence that microbial communities influence host aging trajectories. Researchers will next test targeted microbiome interventions in model systems. From host microbe interactions to physical systems that replicate cell-like behaviors. NYU researchers engineered microscopic oil droplets that alter shape in controlled, complex patterns and engulf surrounding material. The behavior replicates processes normally associated with living cells. Experiments demonstrated tunable responses to external cues. The system provides a minimal physical model for studying membrane dynamics and endocytosis. Further work will explore applications in soft matter engineering and origins of life research, from minimal physical models to automated cognitive testing in wild primates. Emory University and Georgia Institute of Technology researchers created Capuchin AI, which combines facial recognition with real-time touchscreen tasks to study cognition in free-ranging capuchins. The American Journal of Primatology reports a proof-of-concept demonstration of the method. The system removes the need for human observers during testing sessions. It captures individual performance data across repeated trials in natural settings. Researchers plan to expand the platform to additional primate species and cognitive domains, from primate cognition tools to scalable biodiversity data pipelines. The EUB-Cubed project built interoperable data pipelines that automate the conversion of raw biodiversity records into policy-relevant indicators. Case studies tested the system across multiple regions and datasets. The resulting legacy booklet, available on Zenodo, documents the workflow and performance. The tools reduce the manual effort previously required to generate reliable indicators from large, heterogeneous records. Future applications will focus on scaling the pipelines for ongoing monitoring programs, from data infrastructure to precision measurements of exotic ions. Teams at Technical University of Darmstadt and GSI Helmholtz Center for Heavy Ion Research decelerated ions from an accelerator and stored them in a penning trap for the first time. They then performed electron cooling inside the trap. Results appear in Physical Review X. The technique brings highly charged ions to low energies suitable for precision measurements. Future experiments will apply the method to studies of nuclear and atomic properties, from atomic physics techniques to materials properties under strain. A Science Advances study shows that ultra-thin RuO2 films become magnetic when lattice strain is applied, despite the material being non-magnetic in bulk form. Researchers at an unspecified institution grew and tested the layers under controlled strain conditions. The finding resolves prior debates about the oxide's magnetic behavior. It opens routes to engineer magnetic properties in a common metallic conductor and electrocatalyst. Next measurements will examine how strain magnitude and direction affect the strength of the induced magnetism. From engineered materials to astrophysical models of stellar explosions, researchers modeled how small primordial black holes passing through white dwarf stars could initiate explosions matching observed Type Ia events. The mechanism accounts for chemical patterns recorded in supernova remnants and Milky Way stars. Calculations show the events would occur at rates consistent with existing observations. The hypothesis offers an alternative or complementary channel to standard progenitor scenarios. Follow-up searches will look for additional signatures in supernova surveys, from cosmic events to mineral evidence of deep Earth water transport. Brazilian researchers found goethite, a water-bearing iron mineral, preserved inside a 3-millimeter diamond formed deep in Earth's mantle. The inclusion provides direct evidence that the mineral can survive extreme pressures and temperatures. Goethite forms at the surface or ocean floor and incorporates water into its structure. Its presence at mantle depth suggests a mechanism for transporting water into the lower mantle. Additional diamond studies will map the distribution of such inclusions. Before we go, keep an eye on how the targeted immune blockade in interspecies chimeras performs when researchers test additional donor-recipient combinations in the coming months. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time. And if you'd rather watch than listen, find us on YouTube at Nera Network. Links in the show notes. One more thing, if you liked today's episode, our sister show Modern Investing Techniques is worth a spot in your feed. Modern strategies for Canadian and US investors. It's one of the Nera Network's daily briefings, all free at neranetwork.com. The Do Positive Pod is the club for people who do something about it. Episodes and the dispatch wall at neranetwork.com/thedpprod. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.